Hello, this is Dr. Brian Lee. I'm a biochemistry professor at Coastal Carolina University. I would like to invite you to join us this summer to do research in our lab. We are studying the structure and function of protein RNA complexes that regulate the behavior of bacteria found in the food we eat and within our cells. Why do we study protein RNA complexes? When we are infected by disease-causing bacteria, we may experience mild symptoms, such as a sore throat, which gradually goes away with rest and home care. However, these same bacteria can switch from a mild superficial infection to an invasive disease, such as necrotizing fasciitis, also known as flesh-eating disease, which could send you to the hospital or worse. The switch between superficial and invasive infection is mediated by protein RNA complexes. Non-pathogenic friendly bacteria that are found in yogurt and cheese are known to have beneficial effects on our health. They live in our gut and maintain a mutually beneficial symbiotic relationship with us, their host. However, the same protein RNA switch mechanism is also found in these probiotic bacteria, and they regulate how the bacteria interact with us. Understanding the role of these protein RNA complexes in bacteria will help us to treat infectious diseases and to maintain the health benefits of probiotics. What projects will we be doing this summer? We will be working in parallel on both proteins and small RNA transcripts that have been shown to regulate virulence and other metabolic pathways in bacteria. Our past research is focused on ribonuclease Y, which is found in the pathogenic bacteria Streptococcus pyogenes. This summer, we will look at ribonuclease Y in probiotic bacteria, such as Streptococcus thermophilus, which is found in yogurt, cheese, and sourdough bread. Non-pathogenic bacteria are safer to work with, and anything we learn about ribonuclease Y in probiotic bacteria is likely to help us understand its role in pathogenic bacteria as well. Our original interest in ribonuclease Y was based on its role in mediating the switch from superficial to invasive disease during the course of infection with Streptococcus pyogenes. While superficial infections are common and easily treated with antibiotics, invasive infections are often fatal. Although streptococcal infections can be treated readily with antibiotics, resistance to these treatments is a growing concern. Ribonuclease Y regulates proteins associated with virulence by targeting messenger RNA before it can be translated into protein. The KH domain of ribonuclease Y binds to messenger RNA, and the HD domain cuts the RNA, which is then destroyed by other ribonucleases. If we can determine the structure and mechanism of ribonuclease Y, then we may be able to design drugs to block this activity and prevent invasive diseases. One approach to structure determination is X-ray crystallography. Another approach is NMR spectroscopy, which was previously used by our lab to determine the structure of the ZZ domain of another RNA binding protein that's shown here. Our second project for this summer will build on previous work identifying small regulatory RNA transcripts that regulate genes for virulence and metabolism. Previous studies focused on the PEL-SAG-A transcript found in Streptococcus pyogenes, which has dual roles as an mRNA precursor to the neurotoxic peptide streptolysin S, and as an sRNA transcript that regulates several genes associated with virulence. Both roles contribute to the progression of necrotizing fasciitis. Our lab has synthesized several different regions of the PEL-SAG-A RNA transcript that were predicted to contain secondary structure. We have created 3D models of these regions that include key structural features, such as the ribosome binding site and the start codon for streptolysin S, shown here. The 3' end of SAG-A RNA transcript contains another structured region that regulates the expression of the remainder of the SAG operon, which is required for release of the neurotoxic peptide streptolysin S. This summer we will pursue both projects in parallel using probiotic bacteria that we can easily isolate from yogurt and cheese to grow safely in the lab. On the protein side, we will clone the ribonuclease Y protein from Streptococcus thermophilus to begin structural studies by X-ray crystallography. And on the RNA side, we will search for sRNA transcripts in probiotic bacteria that we can synthesize in the lab. We will characterize these RNA transcripts using biophysical techniques and prepare samples for structure determination by NMR spectroscopy. If you're interested in joining us for the summer, please apply through the SCoRE program website. If you have any questions, please feel free to contact me by email. Thank you, and I look forward to hearing from you.